Hello, students. I am recording from my office today because I didn't realize that the other room where I normally record was basically full on Thursdays. So we're gonna make do here. I'm gonna share my screen and we're gonna get going. Hold on a second, I gotta change this. There we go. Okay. Last time we met for lecture, in video land, we had started chapter 10 on muscle tissue, and we had started looking at the structure and function of muscle tissue. We had just finished going through some of the structure of muscle tissue, the different connective tissues that wrap around each of the layers within a muscle, the 4 F words that we talked about, the fascicle, the fiber, the myofibril, and the myofilaments. And we had started to look at muscle physiology. The muscle physiology is kind of how do the muscles work. The anatomy is how our muscles organized. Okay, so it's important that we understand their organization before we can understand their physiology. We had then gone through, started on this, kind of 3 parts here to how do muscles produce tension. Okay, so we initiate muscle contraction through an excitation contraction coupling. We went through some of that last time. And today we're going to continue on the contraction cycling, I should say, or cross-bridge cycling. That's another term for this that you'll hear. And then we'll talk about relaxation. That is, how do we conclude contraction and how do muscles actually relax? So let's speed forward through. We've covered some of this information. This analogy, we talked about how these 2 small molecules, calcium and ATP, are critical for muscle contraction and the roles that they play. And this diagram is kind of a combination of the first 2 points, the excitation contraction coupling, and then cross-bridge cycling or contraction cycling. And so let me make sure that I think you all can see my mouse here, but some cases you can't see my mouse. Make sure that you have a good understanding of the sequence of events that occur from the neuron here, the arrival of an action potential from the neuron to the motor end plate, and how that signal arrives into the myofibril. That is when calcium arrives and starts to bind to troponin. That is all of that from the action potential arriving from the neuron to calcium's arrival in the myofibril. That is all contained in excitation, contraction, coupling. The next thing that we're going to talk about is the cycling. Okay, and that's going to happen in the myofibril, and it's going to happen between actin and myosin. Okay, the sliding filament theory is the relationship between actin and myosin and how they slide past each other, creating shortening of the muscle tissue. And so it's not just that they're sliding, but actually myosin is actually pulling actin towards itself or past itself. And we'll go into some more detail. So we ended up on this figure here. This is the second part here. Let me make sure that I have this going here because I always forget. Contraction cycle. Okay, we have to talk about the beginning, some of these active site exposure, cross-bridge formation, the myocyte pivoting, all these steps. But there's kind of 2 major principles here that you need to understand cross-bridge cycling. And one of them is that binding leads to molecular movement. And the other one is that molecular movement leads to binding. Okay, this creates a cycle. Okay, so I will highlight how this is important, but these are kind of molecular principles about how movement and binding are related to each other. okay? And so the sequence is dependent on these principles here. And let's walk through it. So here's the first, and so each of the sequence of these slides is kind of right on top of each other. So it shows the progression through the sequence here, which will be important for you to understand all the steps. Okay, and you need to memorize the steps. So here we have the actin and myosin in the resting state. Okay, now it says begins here because we begin contraction cycle by the arrival of calcium. Okay, calcium is here in the green, and it is just flooded in from where? Where did it come from? Calcium is stored in the sarcoplasmic reticulum, okay? In particular, into this terminal cisterna. Terminal cisterna is the little bulge terminus of the sarcoplasmic reticulum as it abuts up against the transverse tubule. That's important. So anyways, calcium is flooded out and is arriving here into the myofibril, and calcium is actually going to bind to troponin. Okay, so this next step, we can see calcium is bound to troponin, and that binding, okay, of calcium to troponin causes a molecular movement. Okay, binding causes molecular movement. Okay, the movement in this case is the movement of troponin and tropomyosin off of the actin active site. So upon binding, you can see that that thread of green, the tropomyosin, has shifted. It's shifted off of the yellow little dots. Okay, and that's important because here the dots are covered, and here they're exposed. Okay, those yellow dots on actin are where the myosin head group will bind. Okay, so calcium binding to troponin causes a conformational change, a molecular movement that rotates tropomyosin off of the actin active site. Okay. Next, we have myosin sees its opportunity because it's no longer blocked by tropomyosin, and now myosin is going to reach across and form a cross bridge. Okay, this action of myosin reaching across and binding to actin is allowed by the movement of tropomyosin. Okay, that's called a cross bridge. So I'll come up with, there's an analogy that we're going to use about a tug of war and how the rope and the hands of somebody on a tug of war are analogous to the actin and the myosin. Okay, I'll come back to that in just a minute. But here we can see myosin reaching across and binding to actin. And next comes what we call the power stroke. Okay, the power stroke is the head of the myosin bending, okay, like this, or cocking. And that's where the myosin is doing what it knows what to do best, and that is to grab a hold of actin and then change conformation. So again, the cross bridge happens here, okay? That is, there's a movement that binds to actin, and that movement, the binding of myosin to the actin causes a movement, okay? So here, binding causes movement. And you can see that the red actin filament is now been pulled slightly to the left. Okay, that's called the power stroke. That's just one myosin head group pulling on the actin. So that force is very weak. But if you combine it with hundreds and hundreds and hundreds of other myosin heads, then it becomes strong. So that's called the power stroke. At the end of the power stroke, the myosin head is still stuck to actin because it prefers to bind to actin, and it won't let go unless you have a fresh ATP molecule. Notice that in this position, the myosin head had a hydrolyzed ATP. Okay, ADP is the hydrolyzed form. So you have one phosphate, and then you have the other two phosphates here. They've been hydrolyzed, that is, the bond has been broken, but they're still in the inside of the myosin head. It binds, and then there's the power stroke. Okay, and after the power stroke, ADP is allowed to leave. Okay, notice you can see ADP leaving the head group, and that opens up the head group to bind another ATP. So ATP a fresh ATP can then bind to the head, to myosin head, and we can actually detach from actin. So you notice between these two, you see that detachment of the head. The detachment only occurs if there's fresh ATP, okay? And so we'll come back to this point when we talk about rigor mortis. Rigor mortis is what sets in when there's a dead body and the body gets really stiff, okay? There's a reason for that, okay? So ATP needs to bind, and then the myosin head detaches from the actin, and then it recocks into this ready position. And as long as the actin is exposed, calcium is still present, well, then it will go through that cycle one more time. We go all right up to the first slide. We just start that cycle all over again. Calcium is present. Tropomyosin and troponin have rotated off of actin. Myosin head group is ready to go, binds to the active site there. The power stroke occurs. We slide the filament slightly. Okay, we need a fresh ATP to then cause the detachment, and then repositioning of the myosin head, and now we're ready to repeat the cycle. Okay, so this happens over and over again if we have these two molecules, right? We need calcium and we need ATP. All right, so here's that analogy here of loading the myosin head. Okay, ATP is binds and hydrolyzes to really load that like you would load the spring on a mousetrap. All right, so the combination of all these small events will cause sarcomere shortening and therefore muscle fiber shortening. And you can shorten from both ends, okay? Or if you have a fixed end, let's say this part of the muscle is attached to a bone, okay? This is the real life scenario. That is, one end will shorten and move towards the fixed source. All right, so how do we release tension? That is, once we have produced tension, when does that stop? Okay, so we call this relaxation. It's the third step, okay, because if you go through the first two, but you don't relax, well, then the muscle's permanently in the contracted state. And we actually call that rigor mortis. And we'll talk about more of that in a little bit. So how do we relax? Well, we have this, the duration of the neural stimulus determines the number of free calcium ions. So the presence or absence of calcium is the determining factor here as far as will it continue to contract, okay, given that there's enough ATP. The other issue is, do we run out of ATP? Okay, so if we said these two molecules were necessary, well, it turns out that if we remove them, then we stop contraction. Okay, so that's kind of logical there. So during relaxation, we see that calcium ion concentrations fall in the myofibril. And so calcium detaches from troponin. Therefore, troponin and tropomyosin slide back over to cover the active site on actin. And therefore, myosin can no longer bind, and the muscle will just will release, and all of the filaments will begin to slide back apart from each other. So rigor mortis is a fixed muscular contraction after death, because dead cells and dead bodies do not continually go through metabolism, making fresh ATP. Therefore, that myosin head group is going to bind a hold and then do contraction, and then it won't let go. Okay, the myosin head is attached to actin, and the thing that releases the head is a fresh ATP. Well, if you have no ATP, that myosin head stays connected, and the muscle gets really tough and immovable. And that's why a dead body becomes very stiff. Okay, that stiffness of the muscle and the joints is because there's no ATP to release and return to a resting state. So that's called rigor mortis. All right. So we've gone through these two steps here. We've gone through the excitation contraction coupling, that is from the neuron to the release of calcium. And then from calcium binding, we go through the cycling to produce more and more tension. Okay? At the end of contraction, well, then we talk about relaxation. And it turns out that here in the sarcoplasmic reticulum, we have a number of calcium pumps. That is, calcium is actively, think about like a vacuum for calcium. It gets sucked back up into the sarcoplasmic reticulum. And so therefore the concentration of calcium in the myofibril decreases because it's getting pulled back into the SR. And when calcium goes bye-bye, then tropomyosin and troponin slide back in position, covering the binding site for myosin, and myosin has to let go. All those head groups let go, and the muscle can lengthen again. All right. So tension is produced by these muscle fibers, as I said. Okay, it's a pulling force. There's a number of factors here that determine the type and amount of tension. So the number of pivoting bridges, the fiber's resting length, and the frequency of stimulation are all factors that we'll talk about. Okay, so what are these factors? These factors that influence tension. Okay, what influences tension? Okay, before we get into that, there's a great video that I'm going to put in Moodle, and I would highly recommend that you watch it. And it has to do with muscle physiology. It talks a lot about frog legs and the way that scientists use the gastrocnemius of a frog to determine these relationships between the amount of excitation, the amount of voltage needed to cause an excitation, and the amount of tension produced by the frog leg. And so you can very carefully measure these levels. And a lot of that work is done in vitro, that is in the laboratory setting. Okay, but there's also measurements that we do and assessments that we do in vivo. That is, we do things in the living system. So that video will be posted, and I highly recommend that you watch it to kind of further your understanding of the content that I will give you in these notes. So the first factor that influences tension is this length-tension relationship, okay? A length-tension relationship. And it kind of makes sense. There's an optimum overlap between the actin and myosin, whereby all of the myosin heads that are present are able to bind to all of the actin active sites that are also available, okay? And so if it's too short, not good. If it's too long, also not good, but just right. It's a Goldilocks principle applied to length of the sarcomere. Okay, so let's look at a graph. It's always easier to look at the graph. This graph here, the height of the graph, the y-axis is the tension. Okay, maximum tension is achieved here in this red zone. And the red zone represents that optimum overlap between myosin and actin. Okay, all of the myosin head groups are able to grab a hold of an actin and pull on it. If the sarcomere is too long over in this region, you can see that there's only a few myosin head groups, or maybe no head groups at all, that can actually hold on to actin. So we can't produce as much tension, and therefore the graph goes down on this side. And if the sarcomere is too short, okay, that is, there's not much room for those myosin head groups and the actin to pull, because you've already reached the maximum contraction length, and so you get decreasing amount of tension as you approach that maximum here. So it turns out that the maximum is right in the middle here, and that is the muscle is not fully stretched and the muscle is not fully contracted, but somewhere right in the middle has optimum tension producing ability. All right, so first factor there was the length of the sarcomere. Okay, we call that the length tension relationship. The next important factor that influences tension is frequency. Okay, frequency of stimulation. Now, the neuron that sends the stimulation to the muscle can send multiple signals down the neuron and arrive at the muscle. Okay, and that single threshold signal can induce what we call a twitch. Okay, the twitch is a contraction of the muscle fiber. So this diagram at the bottom is merely to show you, when we're talking about frequency, we're talking about number of events per unit time. This is a higher frequency here, and this is a lower frequency. Neurons operate by this principle. That is, we can have a signal that's like this, or a signal that's like this. Okay? And so the frequency of action potentials determines how excited does the postsynaptic membrane get. Okay, so that's an important thing. We'll come back to that later. So first, we need to understand the relationship again, just between a neuron and a set of muscle fibers. Okay, this is called a motor unit. We'll come back to this definition a little bit later. Okay, it's one neuron plus all the muscle fibers it controls. You can see that neurons branch, and so one motor neuron can control multiple muscle fibers. And in fact, in some cases, it's hundreds of muscle fibers. And so you can have a large motor unit or you can have a small motor unit. And the large or the small is not the number of neurons because it's always one. It's actually the number of fibers and the size of the fibers that makes a motor unit big or small. Okay, we'll come back to that. So the two things that we're going to be comparing here are the stimulus, which is really the activity of the neuron, and we're going to be talking about tension produced. That is the pulling force that the muscle fibers produce. Okay, so one stimulus can produce, a threshold stimulus can produce a twitch. Okay, what is a twitch? A twitch is one contraction. That is one increase and decrease in tension. Okay, and one twitch has three parts. You have a latent period, a contraction phase, and a relaxation phase. Okay, let's look at a graph here. We'll come back to that one. Here, the green arrow always represents a stimulus, okay, a threshold stimulus. So that's the arrival of that signal from the neuron. And then here's time on the x-axis and tension produced on the y-axis. So you always have a little bit of a gap right here. That's called the latent period. And that is, it takes about 3 milliseconds. Notice this time scale is in milliseconds. It's a really short amount of time. That's the amount of time from the motor end plate to calcium flooding into the myofilament, myofibril. It takes about 3 milliseconds for that process to occur. Okay, and then we have a slow increase in tension. That is, the myosin head groups are actively engaging and binding to actin and actually producing the tension. Okay, so we have a growth in tension here, and then we have a decline after the calcium has been sucked out. All of those processes are extremely fast. You can see we've done one twitch in about 40 milliseconds, okay, which is much less than 1 second. All right, so we can see in this prior graph, I'm sorry, I skipped over it. You can see different muscles around the body and the dynamics of the twitch, okay? So the eye muscle, okay, think about the eye muscles that are moving the eyeball or moving the eyelid. In either case, you have very fast twitch, okay? The contraction, shortening and relaxing happens very rapidly. The gastrocnemius is pretty fast for a big skeletal muscle, okay? So that's your calf muscle. And your soleus is more of a walking muscle. It's also on your calf, but its twitch is much longer. It's a more protracted twitch. And so all this to say is different muscles will have different twitch. And the question is, why? Why do different muscles have this different type of twitch? Well, it has to do with motor units, okay? It turns out the soleus has a different arrangement of motor units than the eye muscle. Eye muscle has very small motor units, and the soleus and the quadriceps have really big motor units. Okay, so let's talk about a couple graphs here. There's 4 graphs that you need to know, and these will explore the difference in the frequency. Okay, we said the frequency of stimulus changes the amount of tension. And so in this first graph, we have something called TREPI. Okay, TREPI is a subtle increase in tension with very slow frequency. Okay, so each of these green arrows records a incoming stimulus from the nervous system, but there is enough space between these stimuli to allow a full relaxation phase. Okay, so we increase in tension and then it relaxes fully. We increase it and then it relaxes fully. Okay, so if we increase the frequency, pretty soon we're bumping into this maximum tension that can be achieved with just one threshold stimulus. Okay, that's called TREPI. Wave summation means that now, instead of having these long gaps between stimuli, now we've shortened that. Now the frequency has gone up, and therefore these green arrows are closer to each other, and it does not allow full relaxation in between. We almost relax, and then suddenly it goes up again. Okay, and that happens continuously, and we get this wave summation. That is, over time, they're building tension that was not possible before because the frequency was too low. Okay, this is called wave summation. Next, we have incomplete tetanus. That is, we have a frequency of stimuli producing tension that is approaching the maximum for that muscle. Okay, we're getting to a frequency that is producing this submaximal amount. Okay, and in complete tetanus on the right, we see now the frequency is arriving. It's such a high frequency that now we've rapidly achieved the maximum tension, and it's maxed out. There is no more ability. Every single myosin head group is binding and pulling, and you're producing the maximum tension as possible. And you're going to have fatigue in this muscle very, very quickly, okay? Because every one of those myosin head groups binding and letting go is consuming ATP, okay? It's just consuming it all the time. It's not like it's just holding on with one ATP. No, it requires continuous ATP for that continuous maximum tension. It's called complete tetanus. It's connected to the bacterium that you get a tetanus shot for, like if you step on a rusty nail, the Clostridium tetani, something like, I don't know if it's Clostridium. There's a bacteria that causes tetanus, which causes a locking up of the muscles, and it does it by producing this maximum tension in the muscles. All right, so the reason here for these different muscles producing different twitch dynamics is because of motor units. Okay, so we said a motor unit is one motor neuron and all of the muscle fibers that it controls. Okay, so here this motor unit has how many are there? 7. 1, 2, 3, 4, 5, 6, 7. There's 7 muscle fibers. Let's pretend that they're all touching there, and so that's the size of that motor unit. Okay, motor units can change in the number of cells and the size of the fibers. Okay, motor units can change in the number of cells and the size of the muscle fiber. Muscle fibers are not all uniform in their size. Some muscle fibers are really big and other muscle fibers are small. Okay. So recruitment is increasing the number of motor units stimulated. Okay, so recruitment is increasing the number of motor units stimulated. Let's say I needed to lift a weight that was 25 pounds, and I initiated some stimulus to lift it, and I was like, oh, that's not enough. I can't lift it off of the rack. Okay, and so I recruit a few more muscle group, a few more motor units, and then a few more motor units, and pretty soon I've generated enough tension to lift the weight up off the rack. Okay. Like when you go to pick up like a bag of groceries, you think that maybe the bag is like full of like, I don't know, paper products, like, paper plates or something. It's pretty lightweight. And instead it's like 2 gallons of water on the inside. You're like, oh, like you were expecting something light. And yeah, it was something heavy. You had to recruit more motor units than you're planning on. But eventually your body does that adjustment rather quickly, but it was unexpected. All right. Maximum tension is achieved when all motor units reach tetanus. Okay, so that's maxed out for any given muscle, like the biceps or the quad. Okay, it can be sustained for only a very short time because you lose ATP, and we'll look at the reasons why that is. So here's a diagram showing the different motor units. Okay, these are all of a relatively equal size. But you can see the red neuron is connecting with all the red muscle fibers, and the blue neuron is connecting with all the blue muscle fibers. They're all false colored, of course. They're not really that color. The purple going to the purple. And so over a given task, only one of these might be called on, okay, to do a very simple task, but all 3 of these motor units could be called on for a much more challenging or heavy task. okay, of lifting. The other thing that happens is that for even light tasks, we can cycle through first the red and then the blue and then the purple are all actually trading, handing off their tension producing events between these motor units. And so we produce this smooth tone in muscle, but actually there's on and off happening all the time because the muscle fibers are resting in between. Okay, this only happens in sub maximal tension. So tone. Okay, so muscle tone is the normal tension and firmness of a muscle at rest. Okay, so this is a hard concept for students to understand, but it means just like when you are not actively exerting that muscle. Let's say you're at rest, there's still a low level amount of contraction that is occurring in the muscle because it's just achieving minimal muscle activity just to keep it kind of stable. So postural muscles have high tone. Okay, if you have not been exercising for very much, but you've been upright, that means that your neck muscles are working every day, all day long to keep your head up. Okay, so it's a postural muscle. You have good muscle tone in those muscles. But if you haven't been like working out like with your weights, your muscles, your arm muscles could have poor tone, but your neck muscles could have very high tone. Okay, that is the firmness and the tension that's produced in those muscles during rest. As you increase your cardiovascular fitness and the amount of muscle mass in your body, you have better tone. Okay, that means more muscles are producing a minimal level of tension all the time, and therefore you burn more calories. So better tone, higher metabolic rate. And that's why athletes and people that have bigger muscles and more active muscles, they eat a lot of calories because not only are they working out, but even if they were just at rest, their body would be consuming more calories because their muscles at rest are burning more than a person that has low muscle mass because of muscle tone. So we have to get at the application here. Now that we've understood a little bit about tension, we have to say, well, what are the different types of tension and how we produce contractions? Okay, there's two types, isotonic and isometric, and then there's two subtypes under isotonic. So these are the patterns of tension production we're going to look at. Okay, isotonic means the same tone. Iso means same. Okay, same tone, meaning that we're lifting a consistent weight. Okay, this is an empty coffee cup, but if I'm lifting it, okay, my muscle is always exerting the same amount of tension to lift it because it's only like, I don't know, 1 pound. Okay, it's very lightweight. So the same tone, the same weight. Okay, and so I could be moving my muscles, but it's not ever exerting more than the task demands. And for isotonic, we have two ways there. So when I'm lifting, okay, I could be doing concentric. And when I'm putting it back down, I'm doing eccentric. Okay, so concentric and eccentric are shortening versus lengthening of the muscle. Let's look at a picture here. So here is a muscle, okay, that is lengthened. It's lifting 2 kilograms. So on this graph here in red is the amount of tension produced from rest to now lifting this 2 kilogram weight. And because that 2 kilogram weight is not changing in its weight or mass, therefore this line here is flat. And as soon as you set the weight down, well, then we drop in tension, and that muscle is now achieving 2 kilograms worth of tension. The length here goes from short to long. So you shorten the distance between origin and insertion. Okay, we shorten it and then we put it back down. So you can see the tension produced and the length of the muscle, how it responds. That's how you interpret this graph. So this is eccentric. Okay, so concentric means shortening of the muscle. Eccentric means lengthening of the muscle. Now, think about doing a pull-up, okay? You're hanging from a bar, okay? And you have to pull, like a chin up. You pull your body up, and then your body comes back down. So in no case does your muscle stop contracting. Okay, so you are doing concentric this way, and then you're doing eccentric this way. So think about doing a bicep curl. That is concentric is when the dumbbell comes towards your body, and eccentric is when the dumbbell is going away from the body. Your muscle is still contracting, it's still producing tension, but it's just concentric versus eccentric. Okay. There's more tension being produced on the concentric, but actually eccentric is important as well because you have to maintain that weight in a slow manner as you are lengthening the muscle. So here is eccentric. Okay, we still have, let's just imagine that this support to the 6 kilogram weight was removed, and you still are lengthening that muscle. So you're producing some tension here. You're lengthening the edge of the muscle. And here, this is a little bit confusing. So don't put a lot of stock in the end of this figure here, because the muscle would have to actually tear for this tension to go away, or you'd have to be, the weight would have to be let go of. So concentric, eccentric. Okay, so those are both types of isotonic, the same tone. Now isometric can be most easily described as maintaining the same distance. Isometric, metric is measurement, so the same measurement. That is, you are not changing the position of the muscle, it's not shortening or lengthening, but it is generating tension. Okay, so the analogy I use here is you're holding onto a bucket and somebody is pouring water into it. Your hand is not moving. Okay, so it's isometric, the same distance. You're not changing the position of your hand or your arm, but your arm is producing more and more and more tension to resist the force of gravity that water is exerting inside the bucket. Okay, so we call that isometric. So 2 ways of doing your squats, right? You can do squats in the gym, right, or on the sled, or you can do like a wall sit. Okay, the wall sit is isometric, and doing kind of deep knee bends or squats is isotonic. Okay, so the link is unchanged. So if you were just to remove this a support, you would be holding that weight, but you would not be lengthening or shortening the muscle. All right. I'm going to stop the lecture here. We're going to have kind of a short lecture, and next time we'll pick up and we'll go into these other factors here, and we'll dive into energetics, which is a major topic. See you in class. It's been a while since I used Zoom. I can't find my buttons anymore.